dependent programmed cell death. I guess that's the second part that does show it all. So we looked at these cells in a couple of culture systems. One is a two-layer agar system. Parental cells under these conditions will form spheres of cells very much like solid tumours. The row 0 cells failed to form colonies under these situations. Another assay involves a stem cell, cancer stem cell-like assay, where the cells are grown in serum-free medium with fibroblast growth factor and EGF. And under these conditions, the parental cells formed what are called spheres, small spheres, but they are certainly consistent and high frequency. The row 0 cells failed to form these. However, under conditions of fetal calf serum, both cells grew quite well, although the row 0s were a little slower, as I mentioned. So one of the arguments then is that reactive oxygen species themselves may be promoting this resistance to anoikis that we see in these row 0 cells. And this has been looked at in other cells, and Ross had been associated with this process of detachment-induced apoptosis or reversal of this. So we looked at the intracellular ROS production in these row 0 cells using a mitoSOX marker. The row 0 cells, as we would have expected, produced very low amounts of reactive oxygen species. And this was not only in the row 0, B16 row 0, we have in our lab 6 other human and mouse mitochondrial gene knockout models, and other models also had very low reactive oxygen species. Looking at the peroxide production or intracellular peroxide, again, this was down in each of the row 0 cells we looked at. You heard a little bit about NStyle Cysteine as an antioxidant yesterday. If this is added to the wild type cells, it actually reverses the ability of these cells to form clusters, and the cells curl up and die. So it appears that reactive oxygen species are an important component of the ability of these cells to spread and grow, to invade the local tissue. So in summary, the mitochondrial gene knockout model that I've described is associated with delayed tumour growth in 2 different mouse models, one of them being immune compromised. These cells failed to form tumours in in lung tumours when injected intravenously in both wild type and immunocompromised mice. Failure to form these tumours may be due to this anoikis, or the detachment-mediated programmed cell death that I mentioned. ROS plays a key role in transducing pro-survival signals. It is an important signalling molecule, and it appears to be associated with progression and metastasis, at least in some models. So in conclusion then, the glycolytic tumour model cells have failed to metastasize, and this may be due to events in the early metastatic process involved with signalling due to reactive oxygen species and peroxide. And I guess we're now setting up to look at whether this is a common phenomena in other tumour models. In breast cancer, we're beginning to set up the 4T1 model, and we'll investigate whether or not the mitochondrial genome has similar knockout, has similar characteristics in this model. Thank you.
Thanks.